is the library of all the organisms that the United States has. You can order anything you want, but you have to have a license. Why wouldn't you want anybody just to order what they want? Parents. So they'll send you a live organism in a broth or a slant, or you can send it to them in a live plasma powder. But you gotta have a license. Researcher, university, industry. They won't give it first. Okay, we're going to go on some ways to measure microbial growth, then we'll go back to the phases of growth. Okay? Everybody ready? Okay. Y'all split. We've got people in the back and people in the front. I'll try to be sure I get both of you. Measure bacterial growth. There are different ways to do this. One you already know. Well, measure nucleic acids. Let's get that larger than that. And you've already done this one. I've heard of it. Do you remember Woese? W-O-E-S-E? And he said if you can find the ribosomal RNA sequence, you can find what? Unculturable microbes in the environment. What was the example that we talked about? A little animal that can scratch you. Cat scratch disease. That's one way you can measure is by measuring the amount, meaning the amount of nucleic acids. Okay? Another way is what we're going to do with milk. It's called the plate count method. I'll teach most of this in lab. Essentially what you do is, remember agar? Agar can have, you can actually cool it to the point just before you pour it that you can put things like blood in it, but it won't be destroyed by the temperature, vitamins, or live bacteria. And we're going to do that with milk. In a little while we're going to have a very old milk that somebody took out of the fridge and drank out of it themselves and left out on the snow. That's what kids do. Leave it out for 4 hours and then they put it back in. No, you know you're going to get a microbial load. And then the fresh, clear gallon of milk that people have kept cold, they pour out of it, etc. Well, you take a known sample from the milk or whatever your sample is, give you a hamburger, as long as you blend it up, put it in solution, plate it to plate, so I teach you the dilution series in the lab. And if the plate comes out with 10,000 organisms versus 20, which one would you want to drink? You would drink the one with fewer. It's called a viable plate count. It does require growth. One thing you might want to be aware of is, does the method require growth? Nucleic acids doesn't require growth. You just detect whether they're there. These require growth. And that's the plate count. Another one's called MPN. It's in your book. It stands for most probable number. Most probable number. This is much more statistical. You run a dilution series, you put it into tubes to see if it can ferment a certain sugar, and you count the numbers that grew versus the one that didn't. Okay? Most probable numbers in your book. You can do a series of 5 or do a series of 7 tubes. Some will grow, some will not in a dilution series, and you actually look it up in a book. That's probably on the computer nowadays. That would be a 3-2. 3 grew, 2 didn't. And you have to know the dilution. Read across from the book because they've done the statistics. It will give you a number. They say there are this many organisms per mil. Okay, that will give you a certain number of organisms or bacteria per mil. You have to have growth for that, don't you? Because you knock it out like we did today with the broth. And if they grow, you get turbidity. If they don't grow, you don't get turbidity. Here's the big one. Turbidity. What is turbidity? What's turbidity? Yeah, how hazy the broth looks. The hazier the what? More bacteria. The less hazy the what? Less bacteria. Read this. We read this with machine. You're going to do this in the lab. The spectrophotometer 20. You take a glass test tube, put it down there, a light beam goes across, and it's going to tell you how turbid or not turbid that culture is. Spectro, because you're reading, well, the spectrum, what's a photon? Light. Meter means you're what? Measuring, I don't know why they have 20. I have no idea what the 20 are. But it's to measure light. The more turbid, the more cells, the less turbid, the less cells. And we'll do this in lab. This is a big one. It's used a lot. Now there's a catch with this. As you're actually reading the numbers of the bacteria, they're still reproducing. They're undergoing binary fission. So the best you can do is get a ballpark number, because even as you're reading it, the numbers are changing. Even as you're reading it, the numbers are changing, if you're doing something like Spec 20. Okay, for another one, this is called the Petrov-Hauser counting chamber. Well, take a wild guess what the researchers, what their names were. Petrov and Hauser. You literally have a microscope slide that has a small depression in it. It's actually measured to accept only a certain amount if you feel it right. When you look at this under the scope, you see little teeny boxes. Count the number of bacteria per certain number of box, and you multiply times a known number. One thing you do is you might multiply times 1,250,000, and you will get the bacteria per mil. What if the bacteria are modal? How are you going to count those? Because there's going to be a whipping across there. Then what we did with the protozoans, put them in not molasses, methylcellulose, slow them down. But even then, they're still going to be doing what? Moving. So you're going to have to count them fast. Because even as you're counting them, they're starting to drift from one square to another. Now, this does not require growth, right? Because whatever's in there is what you're counting. A couple of other ones, metabolic factors you can measure. This is based on the logic that the more cells, how fast do they use up food or nutrients? How fast do they produce waste? Maybe acids. And the more organisms, the faster it's going to happen, right? If you have 1,000 organisms, they're going to use up the nutrients and produce more acids faster than if you had 10 organisms. So they're metabolic. Last one is really sort of low-key. Do a dry weight. Get 2 different bacterial samples, or 3 or 4 or 5. Take the same amount out of each, dry them, and they come to like not white powder because they're all colored, but they come to like a powder, like flour. And then whichever one has more weight has more what? Bacteria. Whichever one has more weight has more bacteria. These are some of the methods. This is the big one. If I say, what is the spectrophotometer measurement based on, you say turbidity. You don't say plate count. Turbidity goes back and forth and comes back and back again. And then we'll do a very important thing. That's the bacterial growth curve. And ask questions. I'm not just this isn't just seeing how fast we can get it out, it's understanding. 2 different forms of growth, 2 different ways of growing. Hemostat, batch. The different ways bacteria grow. Hemostat's like the biofilm on a rocky creek. Constantly has water flowing over it, right? So constantly new nutrients or food are brought to the cells. Constantly whatever waste products or acids it produces are washed away. That is a much more active and the more rapid multiplication than the other. Batch is this, is closed. No more food comes in. There's no liquid coming out. So a batch, they're eventually going to use up their food, and they're going to start living and dying in their own waste acids, etc. The bacterial growth curve is based on the batch culture. What did I just say? That means no more food in, no washout of waste. You do cell number versus time. Now, you cannot do this on arithmetic paper. Let me just one thing. You have to do it on semi-logarithmic paper. Semi-log paper.
This is a chemostat batch that you're doing?
This is a batch. This is the batch.
Okay.
The batch. You have to do it on semi-log graph paper. Because what it simply does is it squishes the numbers down. And I have sections. From this section, the sixth section might be a number times 10 to the third bacteria per mil. The next section will have a number, whatever it is, times 10 to the fourth. Then it will have times 10 to the fifth. It compresses. What is 10 to the third? What is that number?
10,000.
1,000. This is the number times itself 3 times. 10 times 10 times 10. 10 to the fourth, 10,000. So you're keeping your units of 10 the same. It's going to squish the curve down. You're going to do this in lab. And I'll explain it there. It looks like this in a classical bacterial growth curve with batch. Everybody okay? Classic bacterial growth curve. Time varies. Every single organism has a different generation time. That means the amount of time it takes to double. E. coli doubles this number every 20 minutes. Mycobacteria might take 3 days, depending. So it depends on the organism. Depends on the organism. They have phases that are caused here. This is called lag. This is called log or exponential. Log stands for logarithmic. This is stationary. You're right, I heard you say it. And death. Cell numbers stay about the same, cell numbers stay about the same, cell numbers increasing, cell numbers decreasing. Okay. Lag is not as quiet as you think it is. Think about it this way. The bacterium used to eat pizza. Suddenly you've jerked all the pizza away from them and they can only eat yogurt. They have to make new enzymes and they have to make new transport proteins to move the food into the cell. They also have to shut down the old enzymes and transport proteins that they don't need anymore. Are they changing the core DNA? No, they're just flipping the switch on and off as to what they need. More realistically, you grew the culture on glucose. Suddenly you took a loophole and you transferred it into a lactose broth. Now the organism's got to make new enzymes for lactose. It's got to make new transport proteins to get things through the cell wall, cell membrane. That's lag phase. Building up enzymes, changing transport proteins, getting ready to really start dividing. Once everything is shifted, you're going to get maximum growth up to the level the DNA can support. That is this phase. That's your highest growth rate. That's where E. coli would be running 20-minute doublings. 20-minute doublings, 20-minute doublings, 20-minute doublings. Now, for some reason, the cell numbers don't keep increasing, but they come to a level where they start becoming level. What is happening, you think, at the beginning of stationary phase?
Food's depleted.
Food's running out, and what else? Waste products are building up. So here you have about, it's not a straight line, here you have about the same number of cells dying as are being reproduced. About the same number of cells dying as being produced, keeping overall about the same number. Now eventually, you're going to be completely or almost, you're going to start depleting your food, you're going to have massive waste products, right? So you're going to have more cells dying than are being reproduced. It's a hostile environment for them. They aren't happy. Don't think that this number, in most cases, will ever go to 0. There's usually 1 or 2 and they're still alive somewhere, or just in stasis, is waiting. Now, there's some antibiotics that are used and they work the best when the cell is actively growing at its fastest level. One of these is penicillin. Penicillin works best when the cell is growing fast because it doesn't break down the cell wall. It never breaks the cell wall down. But if the bacterium is building the cell wall, it interferes with the cross ties, tetrapeptides being tied together. So instead of getting a very solid cell wall, you get bars that aren't tied together, and how loose is that? It's weak. And what's going to make it lyse? Osmotic pressure. Water's going to go in, cell membrane is going to expand, there's no tie downs to hold the bars together, and the exoskeleton is going to blow up. But penicillin works best when the culture is growing at its maximum growth rate. Where would penicillin work best? Log. Penicillin would work best here. Okay, we're going to talk about this. We have Clostridium and Bacillus. What are they? What do I keep talking about them being? They are what formers?
Spores.
Spore formers. Spores are reproductive or survival? Survival. Two of these phases are going to overlap. And the spores are going to come. Okay, deaf and late stationary. That's where you're going to get your spores. Because the organism is running out of food. It's building up toxic products. It's in a harsh environment. It's wanting to survive. Of course, they keep doing it in death all the way. So if you sample Clostridium tetani and you took a sample right here, would you see spores? No. Because it's happy. It's got all the food in the world it wants. They only produce those spores when they're under stress. When they're under stress. This will come back. Guarantee you, that's a major subject. Any questions about this? Okay. Nothing's happening in the lag phase, right? Say no. What's happening in the lag phase? Think for yourself. Engage. Enzymes are being turned on. Transport proteins are being made. Okay. Is that a chemostat or a batch culture situation? If you are maintaining your culture in a chemostatic, always new food in, always waste washed out, where would it be? Where would a chemostat stay?
Stationary
Log. So good, keep talking. It would stay in log. And that's the maximum growth rate. That's where your chemostat would be. Sorry about the clicking. I'm going to go around it. Everybody okay on this? Okay. Before I move on, which is how I started this, is there anything in chapter 6 you would like me to think about before I move to metabolism? Remember, I am doing a review Wednesday. All right, we're heading for metabolism. Let's start this way. I'm in there. The way I teach metabolism is an overview. I don't take you down into the tunnels, but I want you to know, it's going to look like a lot when I finish, but it's actually pretty nice. I want you to understand some interactions and how the energy flows by the time you're finished. Before we even touch aerobic respiration, which is where I'm going to go in a minute, we're going to add a new enzyme for you to learn. The regular old enzyme, which still does exist, just clicking it, that's kind of way around it, okay? Regular old enzyme, nothing special. That's not what they look like. That's a representation. So don't think that all enzymes look like this. What is this called? It fits the substrate. Substrate is what the enzyme works on. It's called the active site. The reason I'm taking you over enzymes is enzymes run metabolism. Enzymes run metabolism. Now, enzymes can do two things. They can be anabolism, and these are in your book too, the words, which I always call synthesis. That means if there are, it can be 1 by 1 or 2 by 2 or 3 by 3, it doesn't matter. In that case, the enzyme will put them together. Will build a larger molecule. Synthesis means building a larger molecule. You always use up energy when you do that. It costs energy to do that. Breakdown, which is catabolism. What do you think that is?
Releases energy.
Releases energy, and it's the what?
Breakdown of compound.
Break it down into bricks, smaller precursor molecules. The term is precursor molecules. The ancients used to do this. After the ziggurat or the ancient city was abandoned, they would go up to the ancient architecture and rip the bricks out and make their homes anew. Same idea. And we probably would do the same thing. Take something already made, break it down into small bricks, and use this to make something else. This may have been part of a cell wall. Maybe you break it apart and you're going to make something else with it. Okay, but catabolism releases energy. It also releases energy. Okay? Back to our enzyme. He's made of what?
Protein.
Protein. In fact, you have a primary sequence. That is how the amino acids actually are in order. The amino acids. What their sequence is. Again, they change for every single protein. It could be isoleucine, leucine, methionine, it could be anything. That's your primary sequence. Later on, once you have these amino acids in their form, we'll go into that later, they will by nature fold, because those amino acids will have only certain hydrogen bonds and they will fold into only one shape. That's your secondary structure. You get to tertiary and quaternary when you start adding all the things together. Two forms of folding of proteins right now. You're going to come back to this later. They can fold like this. That's called beta pleated sheet. They can fold something similar to what you might think of DNA, an alpha helix. And they're not all one. It can be 20% of one, 80% of the other. 50-50, it can be 60-40. Depends on what is needed. Okay, basic enzyme, beta protein. What if I heat this thing up and it's a mesophilic organism and you take it to thermophilic temperatures? What's going to happen to my hydrogen bonds?
It's going to denature.
It's going to denature. What does denature mean?
Loses its shape.
Loses its shape. If it loses its shape, what happens? The active site is changed, and will it function? No. Now some enzymes will flip back and go back to their natural shape when you remove the temperature or remove the acid and hydrogen ions. Some won't. Some are permanently denatured, some are not. That's your core enzyme. Why am I teaching about enzymes? Because enzymes run metabolism. They either make the different structures get rearranged, they may make them get cut into two, they may add them together depending on which pathway you have and what you need. This is a different type of enzyme. There's another type of enzyme that has a remote site called the allosteric site. This enzyme with the allosteric site is called an apo enzyme. I'll show you something else about this in just a minute. You'll get that in a minute. Well, it looks like something fits there, doesn't it? Something's going to fit there.
Is it also an active site?
No, it's not an active site. Active site is still over here. This is called a cofactor. Cofactor. Cofactor can be a metal, it can be something else. It's something that will fit into the remote allosteric site. The allosteric site, if the cofactor is present, affects the shape of the active site. Translate that. If you can affect the shape of the active site, you can turn it on or off. This is a way to regulate or control whether an enzyme is on or off. Two ways you can do this. One is called excitatory. excitatory or apoenzyme. In this case, this is the natural form of the enzyme. Is it turned on or off?
Off.
It's naturally off. To excite would mean, of course, to turn on. So as soon as the cofactor fits in, what do you think it's about to do to the active site? Changes the bonding of the whole molecule to the active site can now fit the substrate. That, of course, means you just did what to the enzyme? Turned it on. Another one is inhibitory, or to inhibit. This, of course, is going to be the complete opposite. The complete opposite.
Is that like when the ATP synthesis stops, like when it gets too much?
You're talking about feedback inhibition. I haven't gotten there yet. That's a different element. That's not this. Okay? Now, for inhibitory enzymes, the enzyme is naturally what? Naturally on. It's the opposite. It's naturally on. This time, opposite occurs. The cofactor binds. What's it going to do to the shape of the active site? Make it wrong shape, non-functional. What did you just do to the enzyme? You turned it off. That's the way that some enzymes are regulated. Here's the other part. The apoenzyme, if the cofactor is in the allosteric site, two of them together, makes a hole, right? Well, the whole thing is called, guess what? Holoenzyme. Called a holoenzyme. Because the whole thing's there. You've got the apoenzyme and you've got the cofactor there. The whole thing's there. Holoenzyme. Now, what was brought up, and it's a good point, is there's another way to control regulation. I'm just going to give you this one. It's called feedback inhibition. And let's step out of the way so you can see. Just have to get written. The whole process is this. You have some sort of compound that through enzyme, say enzyme 1, let's make it enzyme 1, is changed to another form. Through enzyme 2, changed to another form. And you get the idea? Each enzyme performs whatever it needs to do. In the cell, this is going on. Can I erase this?
Mm-hmm.
Okay, erase this. Inhibit, this seems interesting. Yeah, no, move that way. Now, in the cell cytoplasm, or wherever the enzyme is located, if you're measuring, let's say, the amount of D product over time, D is being made and it builds up to a certain level, gets high. With feedback inhibition, D will go back to enzyme 1, turn it off. What happens? Everything's shut down, right? Because D has gone back and either bound to or affected the enzyme further up the pathway. So what's going to happen to the amount of D in the cell? It's going to decrease. When it gets low enough, you're no longer going to have enough D to affect enzyme 1, right? So enzyme 1 is no longer going to be inhibited by the product. It's going to turn back on. What's going to happen? Everything starts back up. D is being produced. What's D going to do? It's going to increase in amount. It can come back. It's called feedback inhibition. Affect the enzyme. Turn it off. What's going to happen?
Everything shuts off.
Shuts it off. And then it keeps going back and forth, back and forth, back and forth, back and forth, back and forth. That's feedback inhibition. So, enzymes depend on the active site having, or the amino acid sequence having the right hydrogen bonding and folding into the correct shape to have the correct active site. Active site bonds the what? Substrate. Enzymes can be catabolic or they can be, well, anabolism. If they're anabolism, are they breaking apart or putting things together? If they're metabolism, what are they doing?
They're breaking apart.
Taking it apart. It's another enzyme that can be regulated by the presence or not presence of a cofactor. It binds at the active site or at the allosteric site. Allosteric site. These are some of the ways you can control metabolism. Some of the ways. It gets more and more and more and more and more of this. Everybody okay with this? Okay, I'm going ahead for the handout of cellular respiration. This is very, very much a diagram. Does anybody need one? Okay. And what's cut off on the side simply says CO2 and ATP for the Krebs cycle. Somebody else need one? I have one. No problem. Don't have them. Everybody okay back here? All right. Okay. Oh, sure. Would you pass them? There you go. Y'all can come up and draw this, right? Say yes.
Thank you.
Anybody else need one? You gotta study. Metabolism, some people think is about the hardest. Some people think genetics is the hardest. And then after that, this sort of thing gets easier, but we'll see. Okay, aerobic respiration. This is where we're going to go. This is one way to teach it. You start off with a process called glycolysis. What does lysis mean?
Break down.
Break it apart. Glyco means, in this case, glucose. Glucose is a 6-carbon sugar. I will ask you to know that. 6-carbon sugar.
So, do you know that?
Yeah, I do know that. Yes, I do. I do want you to know a little bit about that. So, through a series of what? Enzymes. You rearrange it, you cut it apart, you end up, there's a PEP, which we'll go into later, you'll learn phosphoenolpyruvic acid. But just after phosphoenolpyruvic acid comes the thing, pyruvic acid or pyruvate, however you want to call it. This is a 3-carbon compound. 3-carbon. 3-carbon compound. What happened to your glucose? It's cut in two. Now, there's more than that. You rearrange some functional groups, you change some things, but essentially you did what? Cut it in two. This is the core. That's the core. Okay? Now, from this, if you're talking about aerobic respiration, there is an intermediate step. The intermediate step is going to go through a compound called acetyl-CoA. First, you're going to breathe out what you're breathing out right now. Carbon dioxide. Well, you've got to add that extra CoA because there's no CoA on this pyruvic acid. And yes, it's just brought in by enzymes or whatever needs to transport it straight from the cytosol because this is all happening in the cytoplasm. It can also happen if it's eukaryotic in other places. So, coming in is a CoA. You add the CoA, you get acetyl-CoA. How many carbons are in acetyl-CoA? 2.
Because you took off 2.
You took 1 off of 3. You actually have 2 3-carbons, but only 1 3-carbon went here. 1 3-carbon had 1 carbon removed. 3 take away 1 is 2.
Say that again? I'm sorry.
That's right. Pyruvic acid is a 3-carbon. I'm just
Okay.
sorry. You're supposed to ask. You're
Okay.
helping a lot of other people, I promise you. 3 carbons. You lost 1 right here, right?
Okay.
You know, it's floating off in the air. So if it was 3 take away 1 means this is a 2-carbon.
Okay.
We're not finished yet. This is very, very stylized though. There's a system. I just lose my thing.
They dry out.
Yeah. I'm gonna switch to another color, but not for any good reason. Okay, you're green to blue, but it doesn't have any reason. Okay. Series of different structures arranged by different enzymes called the Krebs cycle. And they keep going around and around. Of course, if you need a certain structure, you bleed it off of this and send it over as a Brit to build something else. What it shows on your handout, you have another little CO2 that comes off. You have a small, please understand, small amount of ATP. So I'm going to focus on the electron carriers. What are electron carriers?
Energy.
From going to talk about this. NAD, nickel-adenine adenine diphosphate. He can have electrons added to him in the form of hydrogen, and it becomes NADH. Oxidized or reduced? Oxidized or reduced? Reduced. Which one has higher energy? This one or this one? Which has more energy? Which is the charged battery? NADH. NADH. Okay? That's something we're going to try to keep our minds on. Now, way over the other part, but not really way over the other part, somewhere else, there are a series of proteins, and they can be lipids, they don't have to be proteins. They're always going opposite. This side's reduced, this side is oxidized. This means this has electrons, right? They're in the focus notes. They're flavoproteins, they're ubiquitones, they are cytochromes, and they're metal-containing proteins. Those are 4 classes. Now I can have you learn 20, 30, 40 of each. They're in the focus notes. Flavoproteins, which are proteins. Okay. Metal-containing proteins contain metal. Cytochromes you're probably most familiar with. Yeah, we're close to that one. Okay. And then of course, etc. They pass electrons down. I want you to think of this. They're passing electrons. It's like they're all standing in a line, and one's handing electrons to the next one, handing electrons to the next one in the sequence. If, say, cytochrome, I'm making this up, A hands an electron to cytochrome B, cytochrome B had to be oxidized here, right? It accepts that electron, it becomes reduced. Now he's going to keep passing the electrons. Electrons are how you move energy around. If he passes the electron here to this fella, I have to flip it. This one was oxidized. He becomes reduced. And the electron keeps getting passed. Does that make sense? The very last thing in aerobic respiration, you drop that electron on the final electron acceptor, which means it can't go anywhere else. That's it. That's as low as energy is going to go. Final electron acceptor. Is aerobic respiration, oxygen. How's the electrons being moved? We saw that at NAD and NADH. They're adding electrons on oxygen in the form of what? Okay, you have oxygen. You add electrons in the form of hydrogen. You get water. You get water. I'm not doing these chemical balances precise. It stops. Now, there's a problem. Where do the electrons come from to even be at the top of the dam? Who put the water behind the dam to have enough energy up there to start flowing it down? Glycolysis, somewhat like that, but it goes like this. From glycolysis, you're partially right. You drop it on the top by reducing it, an NADH. So Krebs cycle, you drop another NADH. By the way, this whole system is called the electron transport chain. Electron transport chain. You also drop another electron carrier up, FADH2, flavoadenine diphosphate. How many hydrogens are on this? 2. Which is carrying more energy, NADH or FADH2? Carrying more energy. What are you dumping on the top here? Electrons. You're charging. This is your first massive charging. Then you keep passing it down. Now here's the next part you need to understand. You okay with this? You may just have to memorize it. Now, The electron transport chain is in something. What am I drawing? Where is it? What is it inside? Cell wall. Cell wall is cut to the glycan. Cell membrane. Good going. Don't ever stop answering, and I would never ever get too much down. So inside the cell membrane is the electron transport chain. There, as this is happening, this is back to basic science. There is a compound called hydrogen. Hydrogen, the simplest element is right up here. Hydrogen is one proton with one electron, and it would, yes, it'd be way out there, would be this close in, circling around like a moon. This is hydrogen. Proton has a positive charge, the electron has a negative charge. If it's a whole hydrogen, one positive, one negative mean what? Neutral, no charge. But if you're moving all the electrons around, it so happens that there's something that gets pumped out. And the way you talk about just the proton is H+, because all you have is the positive center. While this is going on, the cell pumps out many hydrogen ions, protons. Many of them. Because it's in the cell membrane. If you have a massive concentration of hydrogen protons outside, this is outside, and you don't have as many inside the cell, what does nature want to do? Make an equilibrium. They want to flow them back in because there's more out here and there's not as many in there. What does a cell membrane block in? Remember, the cell membrane is a semi-permeable phospholipid bilayer. They can't go through the phospholipid bilayer. But there's something called integral proteins. Do you remember those? Can go all the way through the cell membrane, can have channels of pores to allow, depending on the protein, certain things to enter or leave. There is a large integral protein. Guess what? It has a pore in or channel through it. Of course, I could draw it like that. And what are they going to do? They're going to flow back in to try to equalize the charges, both charges and number of particles. This difference in the charges inside versus outside is called the this charge, chemical, because they're actually particles. Difference in concentration, so the next word is gradient. Called the electrochemical gradient has been built up, like putting water behind the dam. You got the water. If you don't have the water behind the dam, you're going to be able to get anything. Now, as this is flowing in, this is what this protein does. Inside the cell, inside the cell, it will take an ADP, adenosine what? Diphosphate, 2 phosphates. It's going to add a phosphate from somewhere inside the cell. And it's going to perform metabolism. And what are you making?
ATP.
ATP. What is ATP? Massive. That's one of your high-power molecules. Yes, it is energy. Adenosine triphosphate. Enzymes, which is also, this is a protein and an enzyme, are often like lipases, ASC is on the end of it. Proteases, ASC is on the end of it. What do you think this thing's called? ATPase. Now, the difference in the charges is called the electrochemical gradient. The entire process of everything being pumped out by the electron transport system, flowing back in through the ATPase, forming ATP from ADP and phosphate, that whole interaction indoor process is called the chemiosmotic theory. Chemiosmotic theory. that's one way you get large amounts of energy through aerobic respiration. Okay, if you're talking about eukaryotic organism and we're flipping, everything occurs with the electron transport chain in the cell membrane. What is a characteristic of a mitochondria in regard to cell membrane? They have the cristae. What are the cristae? Folds of cell membrane. As many as they can get folded inside the organelle. What are they increasing?
Surface area.
Surface area. What are they increasing the production of?
Hydrogen.
What are they? The powerhouses of the cell. This is just free on the cell membrane in a bacterium. Okay, a little bit about electron acceptors. Y'all can draw this, right? Yes. Say yes, you can draw this. Acceptors.
You have to draw it on a test.
No, you don't have to draw on a test. You do have to know questions, and you know my horrible questions. All the following are true except. Okay.
Oh my gosh.
Oh my gosh. Hang in there. You're not, it's not that bad.
Yes, it
Okay.
is.
Final electron acceptor, you can write it, is oxygen for aerobic respiration, which becomes water. Right, that's the aerobic respiration. Oxygen is involved. Okay? Anaerobic respiration is pretty much the same. Everything's a little bit different. Anaerobic respiration means what? Without oxygen. You need to know the acceptors for electrons.
Is this the one on the back side?
No, that's fermentation. We haven't got there yet. We're not there yet. It's not on the handout.
Oh.
This is the same process, but a little bit about the acceptors at the end. This is in the focus notes and on another page. Always ask because you help people. Okay, electron acceptor. This is something to help people understand. Okay, the product. For a compound to be able to accept electrons, it's got to have an empty place for the electron to drop into, doesn't it? It's sort of like if the glass is already filled to the top, you can't put more water in it, but the glass isn't, you can put more water in it, in this case electrons. This technically goes to the SPD and F orbitals and how filled they are and are they balanced, but we're not going to go there. Some electron acceptors, instead of what? Says this is anaerobic. What would be the aerobic electron acceptor? What was the aerobic electron acceptor? Last structure that took an electron in the form of hydrogen, or aerobic.
Is it
Water.
carbon?
Oxygen was the acceptor, became water.
H2O.
Became water, H2O, you're right. Now here, the electron acceptor is what's called sulfate and carbonate. Sulfate and carbonate. There's no way to teach metabolism without having to get you some of this. And this is actually not near what it could be. So, you're going to drop your final electron, dropping through the electron transport chain in the form of hydrogen on sulfate. Your product is hydrogen sulfide. Other one, if you dropped your electron in the form of hydrogen onto carbonate, you can get methane. Methane. A couple thought processes here. Everybody okay? I can slow down because I know this is a lot to absorb.
What is the second one called?
This is methane.
Nothing.
Methane
And H2O is
is hydrogen sulfide.
hydrogen sulfide.
Sulfide. First, if I ask on a test which of the following is able to be an electron acceptor, do you tell me hydrogen sulfide?
No.
No, because what's happening here with the electrons?
It's already accepted.
They're full. They can't take any more electrons. So it'd have to be the sulfate, which can still take electrons. Okay, this one's full. This one's full. All right, a couple of things about this. Hydrogen sulfide, you know what it smells like? Rotten eggs. Very much so. Methane is a flammable gas. Some of you are giving it off. Okay, methane, not all of you, and maybe none of you, it depends on the people. In your intestines, you not only have bacteria that are anaerobic, you can have the anaerobe. What does anaerobe mean when I'm talking about an organism?
No oxygen.
No oxygen, oxygen kills. There's an archaea, so as I say archaea, not bacteria, right? Called methanogens, because they make methane. Some people have them, some people don't. Cows have them for sure. Cows have 4 stomachs. 4 stomachs. One of the stomachs is called the rumen, and it has massive symbiotic methanogens, archaea, bacteria in there in an anaerobic environment. They break down the hay or the grass for the cow to live for food. Cow can't do it. You can put that cow in a pasture of tall, wonderful grass, and he will starve to death if he doesn't have the right symbiotic bacteria and methanogens. So most cows put out methane to some level. Some people are yelling this is causing our greenhouse problem. Maybe, maybe not. Okay, next thing. Can I erase this? Okay. This is sort of related to biofilms. Can y'all see if I erase this part? Biofilms have layers, right? You form a biolayer, what's going to be at the top closest to the environment in the air? Airwaves. What's going to be buried in the bottom? Anaerobes. You go to the sediments of the ocean, sediments and soil, whatever you want to call it. You end up with organisms and they will layer themselves according to oxygen needs. And the biofilm are in the sediments. Top are going to be your aerobes, right? Mm-hmm. The bottom are going to be your anaerobes. Some of the anaerobes this includes are the archaea, the methanogens. What do methanogens do? Methanogens do anaerobic respiration. They drop their last electron on carbonate and produce methane. Methane's a gas. So they're producing methane, and it's going to do what? Methane makes CO2 like a mouse when it comes to the ability to absorb heat. Methane is anywhere from 2 to 4 times better at absorbing heat and holding it in on our planet. So you better be happy this stuff is sequestered in the sediments, because it would make everything we produce from our industrial whatever to be nothing. It would be nothing. But I said they arrange. So right above these, somewhere in there, are organisms called methane oxidizers. They use up the methane. Think about the logic of this. This is the product, and the things finished with this anaerobic respiration, meaning the methanogen. But it is a loaded energy molecule, isn't it?
Mm-hmm.
Loaded with what? Electrons. So another organism is going to come in here, take the electrons off, use them as an energy source, and change the methane to a less energetic form. That's what the methane oxidizers do. The methane never gets to our atmosphere. Never gets. Does that make sense? You see, there's an interaction between biofilm, between final electron acceptor, between anaerobic respiration, and our environment. All ties in. Okay, now there's another one. Are you okay with this? We're heading for the back page. We're going to draw this back. I'm going to draw it very much like this, back to the Krebs. Fermentation. First idea about fermentation are 2. One, it only produces very few ATP. A lot less energy than aerobic respiration. One of the other reasons it runs is we need electron carriers. We need NADH just to run other pathways in the cell. If you run out of NAD, can you make NADH? No, you gotta have the NAD to load it to get to the NADH to help run all these oxidation reduction reactions. What is the combination word for oxidation reduction? Redox. Redox, they just come on words. Fermentation, a lot less energy, functions to actually regenerate NAD, which is an electron carrier for the cell. Fermentation does not have any of this, excuse me for the coughing. No electron transport chain. Krebs is not important because there's no electron transport chain. When I read it, it's death. Well, you get the idea that fermentation might be a little bit simpler. When I erase the electron transport chain, where did most of the energy go? Gone. They can't produce ATP through the ATPase with the electrochemical gradient with the flow back in. There is no electron transport chain. And wouldn't that be a reason why there's a lot less ATP and a lot less energy in fermentation than in anaerobic respiration? Because it's not there. And it's very simple. What I have here are just two samples. You take the pyruvic acid, which you have two. You can do some rearrangements. The one mentioned here is lactic acid. Okay, the other one mentioned over here, there was acetaldehyde. Goes to alcohol. If you ever see abbreviation scientific for ethanol, that's it. Okay, a couple things. I said it regenerates. So what did it just regenerate? Regenerate an electron carrier. It just used up energy though, didn't it? Just used up energy. And coming over to this one, sure, you know, just a CO2 off, but you still have the same thing happening. You're generating what? An electron carrier, but it costs a little bit of energy, a little bit of ATP that comes out. There are hundreds and hundreds and hundreds of individual reactions. You get lactic acid, you can get mixed acid, you can get the alcohol with the ethanol, you get alcohol with isopropyl, you can get acetone. Where do you use acetone?
Nail salon.
Nail polish remover. You can get almost any product you can think of. And we're going to go over that probably in the focus notes. With these products, this is your final electron acceptor. It's a multi-carbon. Here's the big thing about fermentation. A final electron acceptor is a multi-carbon compound of some type. You did not have an electron transport chain, so you don't have ATP from there. Part of the energy is still tied up in that multi-carbon compound. It's never been removed. These products are just the final acceptor for the electrons so they can run some fermentation. Fermentation still lets you live. If there's no oxygen, and you can't run aerobic respiration and you don't have anaerobic, you can switch to fermentation to stay alive. It might give you just a few ATPs, but isn't a few in a life better than a lot and can't use them, you're dead? No, we don't do this. There's an organism in your intestines that can switch from aerobic respiration to fermentation and back anytime it wants to.
E. coli.
E. coli. E. coli does it all the time. Take his oxygen away, he turns to fermentation. He has oxygen, he'll use aerobic respiration. What was a facultative anaerobe? It grows with and without oxygen, but higher numbers where? With oxygen. Why?
There's a hydrogen.
Because it has more energy. It has a lot more ATPs for aerobic respiration to make more cells with than the low ATP production you get from fermentation, which only makes fewer cells. Okay, these are waste products. They have no meaning to the bacterium at all, except to dump that final electron on it. They are not important. Bacterium doesn't care about them. What about us?
We like it.
We like them. We like them. If you get mixed acids, you can get them from Streptococcus, Lactobacillus, there's a list of them on focus notes, which I'll return to in a minute. You can get cheeses, yogurt, you get all sorts of things. Some other fermented products are tofu. I don't actually eat it, I don't know much about it. Soy sauce, okay.